right, so our next speaker is Dr. Sharyl Fyffe Maricich. She is speaking to us today on behalf of her neurobiology background. And I invited her to be here not only because she's a dear friend and I think she's amazing and brilliant, but also because I have heard her speak to parents before at the Rare Entrepreneurial Boot Camp about gene therapy and what gene therapy is. And what I realized is that she has a superpower, and that is to explain really complicated things to an audience in a very simple way that makes you really understand exactly what these drugs and biologics are. And that is not something that I've ever done in terms of explaining the details of gene therapy to you guys, and I don't think I could ever do it nearly as well as she has done it. So I'm really excited for her to be able to really give us 8th grade science class on gene therapy, because for me, that's really where it makes sense, is what is this doing, how is it getting there, and what should we expect from a biological perspective? And so I'm really excited for Sharyl to speak to us today. Her title of her talk is Gene Therapy for Rare Genetic Neurodevelopmental Disorders, the Basics. And just briefly, she has a PhD in human and molecular genetics from the Baylor College of Medicine. She did her training at Baylor College of Medicine in the lab of Dr. Huda Zogby, focusing on Rett syndrome. She currently works at Ultragenyx as the executive director of molecular and cell biology, and she leads a research team of scientists and associates that are focusing on the company's preclinical neuroscience programs, including an AAV9 gene therapy for CDKL5 deficiency disorder. So there isn't anyone better, unless you were developing a gene therapy for Angelman syndrome, that could really explain gene therapy to this audience. So Sharyl, please come up. Thank you.
Good morning, everyone. It's great to be here. Thank you, Allison. That was a lovely introduction. It's tough to follow you. Your talk was amazing, so I'll do my best. So today I'm going to take you through the basics. As Allison said, try to kind of bring it down, simplify it a little bit, and then I'm happy to take any questions at the end. And if you're not comfortable asking me a question in this giant auditorium, please come find me afterward. I'm here. I'll be here all day. I'll be here tomorrow. I'll be at the gala. Find me anytime. We can go sit down. I can draw things out, whatever you need to help you understand. So the outline today is, I'm going to start by just describing what is gene therapy in a very, very basic way, and then take you through the main gene therapy approaches. How do they each work? We're going to talk about AAV. We're going to talk about lentivirus, the advantages and disadvantages of each. And then we'll go through the concept of integrating versus non-integrating vectors, really what's the difference between those two. And then finally, we'll move into how do we engineer a recombinant vector. I'll try to keep this very simple, but I think it is important to understand some of those scientific details. And then how do we deliver the gene therapy to the body? We have a couple of different approaches. We can take an in vivo approach versus an ex vivo approach, and I'll go through those two options. And then how do we deliver the therapeutic genetic material into the cell? So starting very basic, gene therapy treats disease by transferring corrective genetic material to a patient. Gene therapy is a promising approach to correct disease-causing mutations by altering the genes involved. So here what we're doing is the genetic material is typically delivered by modified viruses that cannot replicate or cause disease, right? So that's an important point here. Obviously, we don't want to deliver a virus with something that's going to make you sick, right? So we take the components of that virus away, and we replace that with therapeutic components. So the virus really acts more like a vehicle. Then the delivered genetic material enables the cells to produce a functional protein. And this is an important concept as well. So we're not just delivering the gene, that's not the end. The gene actually needs to be translated into a protein, which is really that functional unit in order to make things better. And the goal of gene therapy is to alleviate the symptoms of the genetic disease and possibly offer a cure. And of course, we never use the word cure lightly. And what I mean there is theoretically or conceptually, if we can get this right, we can get all the details right, this could be curative, right? In a monogenic disease, where we know exactly what causes the disease, we know where that mutation is, if we can put back a functional copy, that could be curative. Now, of course, there are still a lot of things that we need to work through, and particularly for neurodevelopmental disorders, we're really not there yet at that level of a cure, but we're working toward it, and I think that there is room for a lot of hope. And this is a tremendous type of therapy. So before we move on to more slides, I do want to just take you through some very, very basics, and most of you, I'm sure, are very well aware of DNA and proteins and how it all works, but let's just take a few minutes to go through this. So we start with the cell, and within the cell you have the nucleus, where most of the genetic material is stored. And Allison did a great job already talking about the number of chromosomes. So in each cell we have 23 chromosomes from mom and 23 chromosomes from dad. And those chromosomes are really one large continuous DNA molecule. So if you were to unravel that from every chromosome in a single cell and lay it all out end to end, you'd have about 2 meters worth of genetic material. So imagine that, right? You think how small a cell is, how tiny a chromosome is, but all of that genetic material, all that DNA is packed in there very tightly. So then when we unravel that and we look at a segment of DNA that encodes a protein product, that's what we call a gene. The DNA has to be transcribed to a messenger RNA, so it's copied in a way. That messenger RNA is then translated to give you a protein. And the protein, again, is that functional component that you need. So we've seen tremendous progress over 3 decades of clinical trials for gene therapies. There have been greater than 2,500 clinical studies using gene therapy initiated since 1990, so over a fairly long time period, in various disease areas. And you can see those listed on the slide. 3 decades of progress have moved from initial trial failures to trial successes, particularly with monogenic diseases, coming back to that concept of when we know exactly what's broken, it's a lot easier to try to fix it. Clinical trials now are starting to extend to multigenic disorders, so things that are more complicated like heart failure, certain neurodegenerative diseases, but we really are still seeing the most success with those monogenic diseases. So moving on to the main gene therapy approaches, I'll use this slide just to kind of summarize, and then I'll go through each of these approaches in a little bit more detail. So first we have addition, and in this case, you have a mutation that causes loss of function. So you're not making a protein that's doing anything bad for the cell, but it's not doing anything good. It's not doing what it's supposed to do, and so we call that loss of function. In that case, we can just add back a copy of a gene that's healthy, and that cell can then become a healthy cell. For inhibition, we can have a case where either a gene now makes a protein product that is too much, so we have overexpression, or we can make a protein product that's different than it's supposed to be, and that can take on a toxic gain of function, meaning it's doing something different that it shouldn't, and that's toxic to the cell. And so in that case, we would use an inhibitory sequence to shut down that expression and then create a healthy cell. And then finally, we have editing. And in this case, we have a diseased cell with a mutated gene where we come in with an endonuclease, which is something that can bind to the DNA and cut it, and we actually cut out that mutated DNA and we put in good DNA. So this first example of gene replacement therapy, again, here we're treating diseases caused by loss of function mutations that prevent or reduce the production of a functional protein. And a functional version of the mutant gene is added. So we're not taking anything away, we're leaving the cell as it is, it's making a broken protein, it's really not doing anything harmful, we just sort of leave that and we add back in a copy that will be functional. And an example here for all of you would be adding a functional copy of the UBE3A gene. For gene silencing therapy or inhibition, here we're going to reduce the mutant gene expression, right? So in this case, it's predominantly used to treat monogenic diseases due to gain of toxicity mutations, so that a gain of function, something that's new, you actually need to stop that production in order to make that cell healthy again. And that's typically what we see, but an example for the Angelman syndrome community could be using shRNA or microRNA to target the UBE3A antisense. So in this case, UBE3A is not taking on a novel gain of function, but we could enable expression from the paternal allele using this strategy, so using a silencing or an inhibition strategy. And then gene editing, again, is altering the DNA by adding, removing, or replacing base pairs at distinct locations. So here we're working directly on the chromosomal DNA that's inside the cell. And gene editing is based on engineered or bacterial nucleases, things that can come in and cut. And that enables gene ablation and gene correction. And there are 3 main editing strategies that are used. We have zinc finger nucleases, transcription activator like effector nucleases, and CRISPR-Cas9. And I'm sure a lot of you have heard about CRISPR-Cas9. That's talked about a lot. Each of these use slightly different strategies, but based on the same concept. You're using an endonuclease to come to a specific part on that chromosomal DNA where that mutation lies and make a cut. And there are sequences that guide those endonucleases to the correct place. And you can imagine how important that is because you're making cuts in your DNA. You do not want that to start happening at multiple places. And that's still a challenge with gene editing, is really making sure we don't have what we call off-target effects, that we're only bringing those nucleases to the spot of the mutation and only there to cut that out. And it turns out that's difficult to do. All right, so this slide is just for visual learners to really kind of visualize what I've been talking about. And so on the top left, you can see an example of non-integrating gene replacement. So we're taking a gene, we're just adding it, it sits alongside the chromosomal DNA, it does not integrate, it's just an extra copy that lives inside the cell. An example of gene replacement where we are integrating is on the bottom left. Here, we're actually making that cut in the DNA, and we're inserting a new piece into the chromosome. And then for gene editing, we can think about this kind of like cutting and pasting. So the mutation is shown in red. We're going to insert a healthy copy, but we're going to get rid of that diseased copy. So how do we get the therapeutic genetic material into the cell, right? This is obviously not an easy thing to do. And we have both viral and non-viral vectors that can be used in gene therapy. So in gene therapy, a vector is really like a vehicle that's used to bring outside genetic material into the patient's cells. So you can't just put it in, it's not going to effectively just magically make its way into the cell. You need a carrier or something like a taxi. You can think about it like that. It's the vehicle that will bring that genetic material into the cell. In non-viral methods of gene therapy, these can include direct injection or transfection of DNA or delivery using a non-viral encapsulation. And a lot of times what's used is something called a liposome. So this is kind of like fats that will surround that nucleic acid material and help it get into the cell. The viral approaches are where we use viruses that have had their viral DNA engineered or removed, and that this prevents the virus from replicating and causing disease. So now we're going to put in a therapeutic transgene and just use that virus really as the carrier, as a way to get it inside of the cell. So we'll just take a few minutes to talk about adeno-associated viruses, or AAVs. AAVs are small, single-stranded DNA, non-pathogenic viruses. When I say that, I mean naturally AAVs are not going to cause disease for you. So you may have been infected with AAV and you wouldn't even know it. But here, AAVs are engineered for gene therapy to replace viral genes with a therapeutic gene needed by a patient. So we still take away all of the viral genes and we put in our therapeutic gene. They target dividing and non-dividing cells. So this is important for diseases like Angelman, where we really want to target the neuron. So neurons are non-dividing cells. You essentially have all the neurons that you will have in your brain when you were born. And they live with you, and so they do not divide. So it's important that we have viruses that can target non-dividing cells. AAVs are a safer option compared to adenovirus, and this is really due to lower immunogenicity and their non-pathogenic nature. And AAVs are engineered for gene therapy and to be non-integrating, as I showed in the previous example. So just gonna sit alongside your chromosomal DNA, not gonna mess with what's already there. Important terms in AAV gene therapy. I wanted to just take a minute to go through this because as you're navigating through the literature, you're hearing people give talks. These are kind of buzzwords that you hear a lot when we're talking about AAVs. The first one is capsids. So a capsid is really a protein shell that encloses the genetic material of the virus. So that is describing that thing that looks like an icosahedral shape, honestly, a little bit like the light fixtures in here. A serotype is antigenically distinct strains of virus determined by their surface proteins. So as we change the surface proteins on that capsid, that changes the way that that AAV behaves. And there are actually more than 100 different AAV serotypes that have been identified. So you may have heard things like AAV9, AAV5, AAV1, AAV2. These are all different serotypes of AAV. And then finally, tropism is the preference for a strain of virus to infect one type of cell or tissue over another. So we talk about something being neurotropic, meaning that that particular AAV, AAV9 for example, will target neurons, will move its way and deliver genetic material into neurons. And you can have different viruses that are more liver-tropic and so on. So how does the AAV vector get to where it needs to be in the cell? It binds to the cell surface. It is endocytosed. It moves into the lysosome, just different parts that are inside the cell. It needs to escape, and it needs to get into the nucleus. That's where the genetic material is stored. Once it moves into the nucleus, it can then uncoat, right? We don't need that capsid anymore. We don't need that delivery vehicle once the genetic material has made its way into the nucleus. So it uncoats, and you've got your single-stranded DNA. That single-stranded DNA is made into double-stranded DNA. And then it circulizes into what we call an episome. And this is what will exist alongside the normal chromosomal DNA in your cell. So lentiviruses. Some similarities with AAVs, also some differences. Lentiviruses belong to a class of viruses called retroviruses, with HIV-1 being the most widely studied. Lentiviruses have an RNA genome rather than a DNA genome. So remember, AAV's single-stranded DNA. Here you're starting with RNA. And they're engineered for gene therapy by replacing the viral genes with a therapeutic gene needed by the patient. So same concept as with AAV. They target dividing and non-dividing cells, and they have a moderate cassette size. So what I didn't mention on the previous slide with AAV is that you are limited to about 5 kilobases. That's just how much genetic material you can pack into there. Lentiviruses have a slightly bigger packaging capacity. You can get up to 8 kilobases. There are still some genes that are bigger than that. UBE3A is not one of those. It's smaller, so that's good news. So with lentiviruses, because you're starting with RNA, to produce a functional gene product, the virus contains an enzyme called reverse transcriptase, which produces a copy of DNA, or cDNA, from the RNA template. So you gotta kind of work backwards a little bit. You're starting with RNA, in this case, you gotta get back to the DNA to then go forward to mRNA and to protein. So the DNA will migrate into the nucleus. So you have your RNA, you copy it to DNA, that then moves into the nucleus where it integrates into the host genome and provides long-term and stable gene expression. So lentivirus then is an example of integrating. So here it will actually incorporate into the chromosome. However, random integration into the host genome may have unintended effects on the cell's biology. So something to think about. It's great for long-term stable expression to have it incorporating or integrating in that way. So that way, as the cell divides, it will replicate that, and each cell, as it's dividing, will contain that therapeutic genetic material. But you don't control where it integrates, so it can insert anywhere. So you're trying to fix one thing, but you might break something else, right? Depending on where that inserts. If it interrupts a very important gene, you can end up with unintended consequences. So just something to be mindful of. So again, just to hammer home this difference between non-integrating and integrating viral vectors, for typically AAVs are considered non-integrating viral vectors. The transgene stays in the nucleus, it exists extrachromosomally, and the therapeutic effect is diluted by cell division because of that. So as that cell divides, because it's only existing as an episome, it does not get copied, and it does not get passed down to the daughter cells. But because of this, you have a very low oncogenic risk, because you're not integrating. With lentiviruses, these are integrating viral vectors. The transgene is integrated into the host chromosome. You have permanent effects inherited by the daughter cells, and you have this possibility of insertional mutagenesis, or maybe an increased risk of oncogenesis. So how do we make recombinant vectors? Step one is really to just kind of gut that natural virus. We're going to take out all of the genetic material that exists within that natural virus, and we're going to replace it with what we want to be in there. So we do keep some small sequences that are important. Those are, in AAVs, those are called the ITRs. We do maintain that tiny little bit, but the rest of it is removed, and the viral genes can be used later for manufacturing. The second step is to insert our therapeutic gene, and then the third step is to manufacture our recombinant AAV. So to just dial into this a little bit further, I've simplified greatly into one slide a lot of the work that folks on my team do, which is to design this therapeutic transgene, right? This therapeutic vector has a lot of components that are really important, and we take a lot of time as scientists to think about how we structure this. So very simply, you need to have enhancer, and you need to have a promoter for sure, and sometimes you want to also include an enhancer. You can think about these like a green stoplight. It's telling the cell go. It's telling the cell to produce that therapeutic transgene. And these sequences can vary a lot, and they're important because it will tell the cell when to make the transgene, where and which cell types to make that transgene, and how much of that transgene to make, right? So you can use a very strong promoter if you feel like you really are going to need to make a lot of that protein in a cell. Or, as is generally the case in neurons, you want to use a little bit of a more moderate promoter or a weaker promoter, because neurons don't tend to like gobs and gobs of protein being made. It doesn't usually make them very healthy. So it's very important that you consider all of these factors when you're designing these vectors. And then you have your therapeutic transgene itself. In this case, you have a couple options. You can use the native transgene just as it is, make it as natural as possible. You can also do something called codon optimization. In that case, what we do is we make small changes. We're not changing the protein, but we make small changes that can enable the mRNA to be more stable, that can help you produce more protein. And then we have a polyadenylation signal, or a PA signal at the end, and that's kind of like your stop. That's telling the cell to stop, and it's also giving some important stability. So we take that therapeutic transgene and we take that whole cassette and we package that inside of the capsid. So now how do we get that therapeutic genetic material into the body? And here's where we have in vivo options and ex vivo options. In vivo treatment means the genetic material is delivered directly into the person, such as through an injection. So kind of simple in concept, right? And there are a lot of options. You can inject that directly into the muscle, inject that directly into your bloodstream. You can inject it directly into the brain. For ex vivo, which is typically the route that's used for lentiviruses, here the cells are taken from the patient and isolated, and they're treated with the vector in the lab. So this is happening ex vivo, or outside of the body. It's great because you can control which cells come in contact with the vector, because it's all happening inside of a dish in the lab. You can then expand those cells, and then you put those cells back in the body. What can be tricky is making sure that once you put those cells back in the body, that they get to the right place. So that's where it gets a little more complicated. So just to expand a little bit more on the in vivo gene therapy options, when we're talking about treatment for a neurodevelopmental disorder like Angelman syndrome, there actually are quite a number of different options. We can use global delivery through the bloodstream. Capsids such as AAV9 or the AAV9 serotype can cross the blood-brain barrier. It doesn't do so very well. So this is a pretty inefficient way of getting lots of viral vector into the brain. It can be used, but it's not very efficient. It doesn't target a whole lot of neurons. Cerebral spinal fluid delivery allows us to bypass that blood-brain barrier, to get on the inside of the blood-brain barrier. So we go directly into the cerebral spinal fluid that bathes both the brain and the spinal cord. And there are a number of ways that we can access this. The simplest of ways would be through a lumbar intrathecal injection. So we go in toward the bottom of your back, and we insert the vector directly into that fluid-filled space called the CSF that's around the spinal cord. And then it will travel around and travel up into the brain. We can also take advantage of a site of injection that's closer to the brain. It's at the base of the skull. It's called the cisterna magna, so an intracisterna magna administration. In that case, it's a fluid-filled pocket that's kind of right beneath the skull. So you do not need to penetrate through the skull, and you do not need to penetrate through the brain. But it does allow you to get virus very close to the brain. And then finally, we can also do an ICV, or intracerebroventricular injection, which allows you to access that cerebrospinal fluid through the lateral ventricle. So that does require drilling a hole in the skull and penetrating down through the brain to access that pocket of fluid. For the last option here is the intraparenchymal delivery, and early gene therapies targeting the brain took advantage of this approach. It's very good at targeting lots of neurons in a very small space. So if you have a disease where you really are looking to target a very specific place, then that's a great way to do it. But for something like Angelman or for any other neurodevelopmental disorder, where you really want to have global targeting across the brain, you would need to do hundreds of these injections. So it's really not practical. The virus just doesn't diffuse very far outside of that injection site. So how does the vector get to where it needs to go in the brain? And this is just kind of a picture taking you through the whole process. And if you're a visual learner, hopefully this is helpful. So here I'm showing the example of an intracisterna magna injection. So again, at the base of the skull, you can see shown in blue, this is the ventricular system. This is where the cerebral spinal fluid will circulate. The vector goes in, it makes its way through into those lateral ventricles, and then it diffuses out into the brain parenchyma. So that's shown in gray. Once it targets a neuron that lives within that brain parenchyma, it is then endocytosed, and it moves through, it moves into the nucleus. You have your mRNA, and then you have your functional protein. And a major challenge with AAV gene therapy, I should mention, is the targeting of a sufficient number of neurons in a large brain. And this is something that my team has been working very hard on, is really trying to optimize how we do those delivery conditions. Is it, what do we put that vector in? Is it a solution that makes it heavy? Are we injecting very quickly? Are we injecting slowly? There's a lot of details that are being worked out to really optimize how that AAV can travel throughout the brain and contact as many neurons as possible. So just to start to recap here, I made this slide to outline some of the key advantages and challenges of both AAV vectors and lentiviral vectors. There are many more things than this, but I tried to distill it down into something that could fit onto one slide. So for AAV vectors, some of the advantages are that they are ideal for in vivo delivery. They're ideal for targeting non-dividing cells like neurons. They have a very low integration rate. So we talk about AAVs as being non-integrating, and for the most part, they are. Occasionally, there is a few integration events, but it's very, very, very low. And because of that, we have a reduced risk for oncogenesis. And there are multiple capsid choices to tailor your cellular tropism. So those are those words that I talked about earlier, meaning that you have a lot of choices of AAVs depending on where you want to target. AAV9 is commonly used, particularly if you're going to go into the cerebrospinal fluid and target the brain. Some of the challenges are that they are limited in their capacity. You could only fit so much genetic material in. So if you have a very large gene, AAV may not be an option for you. Another challenge is that you're limited to a single dose. And actually, this can be a challenge. It can also be an advantage. If you can target enough cells, maybe you only need a single dose and you're all done. That's an advantage. However, if you're not targeting all the cells in the brain, right now, we're limited to a single dose, and that's because once you've dosed with AAV, your body will make neutralizing antibodies against that AAV, which prevents you from redosing. And there are folks working on strategies to enable redosing, but we're not there yet. And so that comes to that next point with pre-existing neutralizing antibodies. Maybe you had a natural infection with AAV at some point in your life that you didn't know about, and you made those neutralizing antibodies. That can make you ineligible from treatment with AAV vectors. And then the last challenge here is that there are lower numbers of cells targeted in the brain, and the expression is not always stable. So we're still working out the kinks. It's getting better every day, but we do still have some work to do there. For lentiviral vectors, these are more ideal for ex vivo, right? Take the cells out of the body, treat them, and then put them back. They're ideal for targeting dividing or non-dividing cells. They infect a wide variety of cell types. Therapeutic effect will not be diluted by cell division. That's one of those advantages of having that integration event. And there's relatively high numbers of cells that are targeted in hematopoietic stem cells. Challenges are that there's a medium genome size, so it's a little bit better than AAV, but it's still limited. Lentiviral integration poses a higher oncogenic or a genotoxic risk compared to AAV. So we don't know where that's integrating, and that could sometimes cause a problem. And potential activation of neighboring genes post-integration. So this is all part of that problem. You can't control exactly where that goes into the genome. You are limited to a single dose, so similar to AAV. And corrected cells need to travel to the brain across the blood-brain barrier. So when we're talking about hematopoietic stem cells, we treat those in the dish, we put them back into the body. If the target is the brain, they still need to target up into the brain and release UBE3A so that can be therapeutic. So as we start to wrap up here, I'm just going to take you through the key steps in gene therapy, sort of from beginning to end, that will hopefully bring together most of what we talked about today. So step one is to identify a therapeutic gene and design the vector and the delivery strategy. In step two, you insert the therapeutic gene into an appropriate vector. In step three, you deliver the therapeutic gene or the vector to the patient, and that can be done either in vivo or ex vivo. In step four, the vector delivers the therapeutic gene to the nucleus of the cell, right? So it's not enough just to get it in, it needs to get into the cell, it needs to get into the nucleus, and then it needs to make a protein, which is step five. And can we really do this? Well, I'm excited to say that yes, we can. There have been approved therapies for AAV and lentivirus products. Not a lot, but there's a handful. And there are many ongoing trials, and so you'll see this continue to grow over time. So the key takeaways for today are that gene therapy has the potential to dramatically improve genetic neurodevelopmental disorders by targeting the underlying cause of the disease using a one-time treatment, and that's pretty exciting. There are successful FDA-approved examples with recombinant AAV and lentiviral vectors, as I just showed you, for some diseases, but nothing yet for neurodevelopmental disorders. So we hope to see more of this to come. Each gene therapy strategy and delivery vehicle has its pros and cons. There isn't a one-size-fits-all. There's not a magic answer which one is best. I can't tell you that. I think it really depends on a lot of factors, and that's why it's so important, as Allison has mentioned, that everyone is as informed as possible. So ask lots of questions. Success is critically dependent upon a deep understanding of the underlying disease biology and clinical presentation, as well as a strong non-clinical package. The determination of optimal gene therapy or gene delivery parameters. So optimizing that route of delivery, the dose that you're giving, the formulation, which is what that vector is, what kind of liquid it's in. All of these things are important to maximize the number of neurons that take up the vector across the brain. Success is also critically dependent upon the ability to manufacture the vector in large quantities. We don't always think about that early on in the research world. It's easy to make enough vector to treat a mouse, right? It's very tiny. But when we're talking about treating human beings, we need to have a way to scale up that manufacturing process. And then development of appropriate clinical outcome measures and robust objective biomarkers. And FAST has done such a great job in this area. We really need to work on that so that we can capture key aspects of the disease and how they change over time, right? It's one thing to go through all the science to get there, but we have to be able to measure it in the clinic. And then finally, optimal immunosuppression protocols need to be determined to limit unwanted or detrimental immune responses that could limit efficacy and durability. So at the end of the day, these vectors come from a virus, right? So our body is going to react to them in some way. So even though we've taken out all that viral DNA and we've inserted our therapeutic transgene, we still can have these immune responses, and we have drugs that we can use to limit those immune responses. And there are lots of folks that are working on figuring out what does that optimal immunosuppression regimen look like. And that's all I have for today. So if we have any questions, I'm happy to take them. Maybe we have some time. And if you're not comfortable asking questions in this forum, please come find me at any time, and I'm happy to work with you.
Thank you very much. I was curious if you were saying that there are some prior therapeutics that may affect the other therapeutics that might go on top of it. What about mRNA and, for instance, the COVID vaccine? Do you think that has any impact on the efficacy of any of these therapies?
That's a great question. So I think what we, particularly with AAV, what we really need to be careful of is something that is going to look like that AAV. So if the body has seen that before, even sometimes you can have cross-reactivity between different serotypes. So you could have somebody who's been infected with an AAV8 that then would not be able to have an AAV9 because they have neutralizing antibodies. In my understanding, that's going to be different, and so it wouldn't, at least from getting an AAV gene therapy, those COVID vaccines would not make you ineligible.
It sounded like one of the things you touched upon is an undesired, that these can have the desired effect, but they could also have potentially an undesired or you kind of briefly touched on that, and I'm wondering about that. It sounds potentially problematic.
Yeah, thank you. That's a great question. So that, what we're typically talking about is in the case of integration. So when we are having a random integration event, like with lentivirus, it's random, right? And so you don't know where in that genome it's going to disrupt what's already there to insert what you want, right? That's the healthy thing that you're putting in. But as you make that cut, if it happens to be in a gene that's critical for something else, you've now disrupted that gene. So it doesn't work anymore because you took it in half and you opened it up and you put something new inside. Now there are lots of places in the genome where you can make those cuts and nothing bad will happen. But it's random, right? And so sometimes it can, and it's important to monitor kind of where those integration events are happening.
I have a question. So when you take the virus and you take out the bad virus and you put the right serum in, it's not a serum, but a protein, I think. Mm-hmm. You put the correct one in. Is it the same solution in there that for, let's say, every mutation? So is it like a one and done for all mutations and a one and done for all
Great question.
deletions?
Great question. So In the case of sort of a simple gene replacement where you have a loss of function, and it could be lots of different mutations that cause that, right? Different point mutations or deletions, anything that causes that protein to either not be produced at sufficient levels or not be produced at all, or basically just not work. In that case, what you would do is design a vector with the whole copy of a brand new gene that works. You're gonna put that whole thing in. And that's just gonna live alongside the other broken one. So it could be broken in a lot of different ways as long as it is causing a loss of function, meaning it's just not working. And so then you put that back, and that would work for a number of different folks with different mutations.